Hello, class. This is your professor, Professor Daryl Omar Fisher, here to continue on with our studies in biology, specifically speaking to Chapter 4, the energy of life. Remember that the consumption of energy is a sign of life. And so things that are alive need energy so that they can continue to operate and to live. And so let's study that in this chapter. All right. So in the picture here, you have somebody riding a bike that requires energy. Where does that energy come from? Primarily, it comes from the food that we eat, right? So many of the biker's cells are extracting energy from the food and using it to power his muscles. I see this biker is very muscular. Look at his legs, very muscular. But that energy has to come from somewhere, right? He has to. If he stops eating food, he won't uh, not be able to do things immediately, but eventually his body will run out of energy, literally run out of energy. And we call it starvation. But starvation is lethal because your body needs energy. But what is energy? And as you see here, it's the ability to do work, the ability to do work. We have energy that gives us the ability to do work. Two different types of energy. We talk about kinetic energy, sort of energy in action, and potential energy or stored energy. Right. So you look at a spring. If you push a spring down, it has a lot of potential energy, potential to do work. So it's stored energy. Once that spring has is extending, is releasing, now it becomes kinetic energy, energy of motion, movement. Right. Very similar. We have uh, chemical bonds, which are potential energy, right? Chemical bonds. So if we look at different substances, we have chemical bonds. Remember, talk about um, we talk about organic compounds, and we talked about triglycerides, fats, fatty acid chains. There are carbon bound to hydrogen. We have energy in those bonds and those electrons that are being shared or somehow bond together. Well, if we can break those bonds, we can release energy that can allow us to be able to do work. So, to release the energy, the cells break bonds, and the cell can capture that energy. It can use that energy to work. If it doesn't capture that energy becomes lost as heat. Energy is converted from one form to another. So the idea is energy is not created or destroyed, but it's simply converted. It's not created or destroyed, it's simply converted. So energy from the sun gives photosynthesis, lets the tree grow, provides food for the animal through cellular respiration. They can make ATP and it can power their muscles and releases heat, water, and the cycle continues to happen. That so we have energy being just converted from light to sugar to ATP to powering muscles to movement back to heat, light, all these things. All right. And that's how we get transformation of energy, conversion of energy. And it is, as you see here on this slide, it is inefficient at times. Energy is lost as heat, right? That doesn't mean that it disappears in the sense of never to be found again, but is not being used in a converted to a useful form of energy. You exercise, your body gives off heat. Uh, that heat is not necessarily being used as further energy. It is being lost. And so uh, we want to definitely pay attention to that and to understand how we can use energy, utilize it, and um, keep it as much as possible. And our bodies are have ways of utilizing energy, but definitely have um, also ways of. So it takes a lot of energy to stay ordered, to continue through our life processes. And so cells are constantly using energy to build molecules, structures, perform chemical reactions, make proteins, as we've talked about, um, do the different things required to be able to sustain life. Metabolism includes all chemical reactions. So our bodies are made up of, as we talked about, atoms, molecules, different reactions constantly happening throughout our bodies, right? We talked about H2O, we talked about glucose, we've talked about fatty acids, we've talked about nucleic acids, we talked about oxygen, carbon dioxide, some of the different things that are inside of our bodies and are being made or used in our bodies. And so our bodies are these complex chemical reactions um, to build even more complex molecules out of monomers, out of simple molecules. Um, to build a chain of amino acids to make a protein that requires energy to break down molecules. So to break down a chain of some type of monomer, to break it down, you can release energy, right? So we have different types of reactions that are happening. Endergonic, as you see here, reactions that form bonds to build molecules require energy. And so the energy of the products is higher than that of the reactants. So you have two things you're bringing together, requires energy, endergonic, needs energy to go in for that reaction to happen. An exergonic reaction is the reverse. Generally, it's a reaction where you break a bond. So we have something, the bond breaks, releases energy. All right. So a reaction that releases energy is an exergonic reaction. And then you have most energy transformations which occur are in oxidation reduction reactions, and that's transferring electrons. So to oxidize is to donate electrons. Remember, electrons have a negative charge. Protons have positive, neutrons have a neutral charge, electrons have a negative charge. So if I donate electrons, if I lose electrons, I've been oxidized. That's the terminology that we use. But if I accept electrons, if I'm the one accepting the molecule accepting electrons, that means I've been reduced, because if I you accept negatively charged electrons, negative charge, the more negative, the more I am reduced. Think of math, a math equation. The more negative you get, the lower you get. So that's kind of how you could remember that. So, all right. So, oops. Sorry. What did I do? Oxidation is the loss of electrons. Reduction is the gain of electrons, which requires energy, right? And then we have these oxidation reductions that it can occur simultaneously, and that's called redox. And so we have this, what we call in our bodies, an electron transport chain. So an electron being constantly moved down a chain, a series of reactions that are happening by these electrons being moved down a chain, therefore rele- releasing more and more energy as it's being moved from this one and then being released and moved to that one and then being released and moved to that one. And we call that an oxidation, linked oxidation reduction reactions or redox reactions, right? And so ways we can sort of maximize the energy, photosynthesis and cellular respiration. So photosynthesis happens in plants, cellular respiration happens primarily in animals. We see that as their primary source of energy. Both use electron chains. So you form a bond with an electron, break that bond, release energy, form a bond with another protein, break that bond, and keep that happening further and further down a chain. Adenosine triphosphate, also known as ATP, is a nucleotide that temporarily stores energy. It is the form of energy that cells can use. Think of ATP as like the gasoline of the cell, right? It's sort of it's a molecule. It's a nucleotide, but it's a molecule that has a three phosphate group on the end, and that triphosphate group, that last phosphate group, has a bond with a lot of potential energy. And if we break that bond, we can change that potential energy into kinetic energy and use the energy from the ATP. So removing that endmost phosphate by hydrolysis. Whenever you see the term lysis, you're breaking something. So in this case, hydrolysis, using a water molecules, and you see the H2O there. The water, you need a water molecule to make this work. You need water to be able to create energy. It's another reason why drinking water is very important. But you need a water molecule for this reaction to be able to happen. You break off that last phosphate group, release that energy, and go from ATP, adenosine triphosphate, to ADP, adenosine diphosphate. ATP is formed during cellular respiration, so a series of chemical reactions that release energy from sugar, the food that we eat, helps producing ATP from ADP. So we eat energy, and our body can use that energy to take ADP, adenosine diphosphate, add a phosphate to that, and make ATP and store energy in our bodies using the energy from the food that we've eaten. ATP breakdown, when our body uses this ATP, uh, happens, we couple it with a endergonic reaction. So a reaction in your body that needs energy will then use the ATP that's been stored up, which has been formed from the energy that you've eaten, and it'll couple that with a reaction that needs energy in your body. All right, so this is giving you an example. Um, ATP energizes target molecule, making it more likely to bond with other molecules, right? And this is forming a polysaccharide, right? So the ATP will help that uh, those glucose molecules bind together and make a longer polysaccharide. And so bonding the monomer, the monosaccharide glucose, bonding glucoses together requires energy. And so the ATP will give it that energy so that it can bind to other glucoses and form a longer polysaccharide. This is showing you another way ATP can work. Donating the phosphate group changes the shape of a target molecule in the muscle cells. ATP will change the shape of the target molecule involved in the muscle shell as well. causing it to be able to move and to work, and therefore your muscles expend that energy. Next, we have the enzyme. Enzyme is very important when it comes to different reactions happening in the body. An enzyme is a protein. Okay, we talked about proteins. Remember proteins, lipid, nucleic acids, carbohydrates. Enzyme is a protein. Where are our proteins coded for? Remember, they're coded for in your DNA. So an enzyme is a protein that acts as a catalyst. What is a catalyst? A catalyst speeds up a chemical reaction without being consumed, right? So you need something, a reaction to happen, uh, and the protein, the enzyme, that is a protein, it's an enzyme, helps that reaction to be able to happen. And you see a simple example here. Um, a more complex molecule comes to this enzyme substrate. The purple object is the enzyme that binds to the enzyme, and then the reaction, the exergonic reaction, the reaction of breaking it down, can then happen. Right, so substrate binds to the enzyme's active site. Once chemical reaction occurs, products are released, but the enzyme retains its original form. It's a catalyst. So the enzyme is not part of the reaction. It just helps lower, it helps the reaction happen, lowers the what is called activation energy. And that's on this slide. So the activation energy is the energy required to start a reaction. Without an enzyme, it takes a lot of energy to start a reaction. But with an enzyme, you can lower that activation energy and make it easier for that reaction to happen. And so that can be, you can think about that. There is right now, where you are, wherever you are, there is oxygen and hydrogen floating around in the air. But why isn't it becoming water? There's hydrogen and oxygen. Hydrogen and water is simply H2O. So why isn't the hydrogen and oxygen that is floating in the air that you're breathing right now, why isn't it just coming together and coming water? Because it takes energy for that to happen. And it won't happen on its own because the activation energy is too high. But what enzymes do is allow certain reactants to be able to react by lowering their activation energy. So making it more likely that they will react and make the reaction happen. Enzymes also have cofactors, which help participate in the reaction and further increase the enzyme activity. So metal ions and vitamins are common cofactors that help certain reactions happen in your body. Only certain chemical reactions are happening at any particular time in your body, right? So we don't want every single reaction that your body can do to be happening all the time. But the enzyme may be in your body, or and the different reactants may be in your body, but we want to sometimes inhibit and not allow certain reactions to happen at certain times. And so we can, in your body and plants and animals, that can happen. We can have enzymes be inhibited. So that enzyme uh, has an inhibitor that stops it from being a catalyst for the reaction. And your body can therefore control, okay, we don't need that reaction to happen. Let's inhibit those enzymes. Or when we do need that reaction to happen, let's remove the inhibitors from those enzymes. Okay. And that's talking about how it can cause the inhibitor can cause the enzyme to change shape and therefore prevent the reaction. Cells can use a negative feedback to control biochemical reactions. Right? It's a process called negative feedback. The product of a reaction slows the production of more product. Right? So negative feedback. I have a reaction. For an example, I need to uh, make some. I need to make some molecule in the cell. Um, Negative feedback says uh, the more I make, the more it's going to tell the cell to not make anymore. So if I uh, if I need more, hemoglobin for my bloodstream, I make more hemoglobin, which is the hemoglobin, and it is it binds to the oxygen that you breathe in so that your blood can carry oxygen to your body. Let's say my hemoglobin levels are low in my bloodstream. So the negative feedback mechanism in your body causes you to make more hemoglobin, but then as you make more, it's going to tell your body, okay, you're making more. So now let's slow the process of making hemoglobin because we don't want to make too much. That's negative feedback. The more I make, the more it tells my body to stop making it. But you can also have positive feedback. The production of a reaction stimulates its own production. So certain things, if I make more, it tells if I have a positive feedback, if I make some of it, it tells my body to make even more of it. That's a positive feedback. Most things are negative feedback because we want to stay nice and balanced. And if we've gotten out of balance, we want to get, or out of our range, we want to get back into balance. Temperature affects enzyme activity. Temperature simply affects reaction activity in general, right? So um, remember, enzymes are proteins that are catalysts for specific types of reactions. Right? Proteins are affected by pH. Proteins are affected by temperature. Proteins are affected by concentration as well. So different things because these affect proteins, they will affect enzymes, and therefore they will affect reactions. So most enzymes are active within a range of temperature. Remember, if at too high or too low, proteins can denature, change shape, and therefore not be effective as a catalyst for their reaction. Right? If I change the shape of the protein, it's not as effective. Therefore, and that can happen based on temperature. Enzymes also have optimal salt concentrations and pH. We just talked about that. So, also, if a if we know that our bodies have a certain pH, if the pH changes too drastically, it can also cause the enzyme, which is a protein, to denature and therefore change shape and therefore not be effective as a catalyst. Now let's get to the cell membrane. The cell membrane we talked about is a structure that surrounds the cell. Phospholipid bilayer with embedded proteins, carbohydrates, lipids, and other things embedded in it. But its purpose is to surround the cell, provide a barrier, protect the cell, be selectively permeable, decide what goes into and out of the cell, all the things. Why is that important? Well, we want to talk about gradient. We want to talk about concentration gradients. Right, and you see it here Everybody has experienced this. I'm sure you've used a tea bag. Well, put a tea bag into water, and the tea not naturally diffuses, called diffusion, diffuses to the water from high concentration to low concentration. Same thing happens if you put um, some red dye into a glass of water. Eventually, the whole cup becomes pink. Why is that? Because that dye has diffused, it's naturally spread out through the water. It's moved from a high concentration, the droplet, to the low concentration, the rest of the cup, and it naturally diffuses throughout, right? So that's what happens. The example given here is tea, right? So it has a tendency to dissipate, to diffuse, to move. So we also have transport across the membrane, right? So we just talked about diffusion, um, but there's several ways you can have things transport across the membrane. Remember that the plasma membrane is like a fluid mosaic model, like a jello almost. It's very fluid. It moves. It's not stagnant, right? So passive transport, that means do not require energy, moving down a concentration gradient. So moving from high concentration to low concentration. Um, you have simple diffusion, which is, think of the tea bag, think of a drop of dye in the water. That's simple diffusion, high concentration, low concentration. So something on the outside of the cell that is at a high concentration could diffuse into the cell where there's low concentration. Osmosis, that's the movement of water. So water can move based on the concentration of the whatever is dissolved in the water. And then we also have facilitated diffusion. All right. So these things happen sort of naturally. But then you also have active transport, which is transport that requires energy. And you have um, transport in vesicles by endocytosis or exocytosis. So pinching off portions of the cell membrane. And so these also require energy. So there's several ways things can be transported into and out of the cell. Right. So passive transport, I just mentioned. One of those is simple diffusion. So when it occurs when the concentration gradient dissipates across the biological membrane. So things naturally move from high concentration to low concentration. Only small nonpolar molecules can cross membranes by simple diffusion. So not everything can go into your cell by simple diffusion, but small molecules, nonpolar. Remember those tails of that phospholipid bilayer are nonpolar. So those small nonpolar molecules can cross the cell membrane's hydrophobic barrier. Passive transport also includes osmosis, which is the diffusion of water across the membrane, right? So certain things in the water, uh, based on the concentration of whatever is in the water will move across the membrane to balance out the concentration of the solute in the water, right? So on outside of the cell, I have a lot of glucose in the water. Inside of the cell, I have a little bit of glucose in the water, right? So water will go out of the cell to dilute the high concentration so that now it's balanced, right? And we have equal concentrations of solute in the water. And so this is kind of showing you that osmosis is driven by the concentration gradient of water. So when a blood cell is in an isotonic solution, this is why when they give you saline, when they hydrate you in the hospital, they give you saline, not just pure water, because they want it to be similar to what is in similar concentrations to what's in the body. So if your cell is in an isotonic, same concentration, that's what isotonic means, there will be no movement of water in or out, or an equal amount of movement. If you place a blood cell in a hypotonic solution, hypo less than, water will, so that means it has a higher concentration of solute inside the cell than outside the cell. Water will come in and try to dilute that. And then if you have a hypertonic solution, the reverse happens. That means you have a higher concentration of solute outside the cell, hyper, more. And so water will come out of the cell and try to dilute to balance out the concentrations of the solute in the water inside and outside the cell. Right? And so that's what's happening there. Equal concentrations, higher concentration of solute inside the cell, lower concentration of solute inside the cell. I mean, or you could just say higher concentration of solute outside the cell. Osmosis determines water content in plant cells. So plants usually keep the solute concentration inside their cells lower than the outside so that water enters the cell. They want water coming in. So they want a hypertonic surroundings resulting. Sorry, plant cells want to keep the solute concentration inside the cells lower. So they want the concentration of solute outside higher. Um, lower down so that water enters the cell. So water enters the cell. That's what we want. Hypotonic hypotonic surroundings. So we want water to come in and try to dilute the inside. Therefore, water coming into versus going out of the cell. Facilitated diffusion does not require energy as well. Another type of diffusion, when the membrane proteins transport across the cell. So something may not be able to diffuse through the plasma membrane per se, but remember those proteins that are embedded in the plasma membrane. Some of those proteins are protein channels. And some of those channels, the substance can diffuse through the protein channel, maybe not through the phospholipid bilayer, but it can diffuse through the protein channel, but it's still still moving from high concentration to low concentration. So it still doesn't require energy, even though it's going through a protein channel. Right, so that's talking about that. Now, active transport occurs when membrane proteins use energy, active energy, and this type of membrane transport is often called a pump. You'll hear it called protein pump, something like that. Substances moved against their concentration gradient. So things will naturally move from high concentration to low concentration without any energy. That's just natural movement. That's how nature works. But if you wanted to go from low concentration to high concentration, and that's different, that requires energy. If I wanted to push against the concentration gradient, you know, if somebody farted, for instance, it's natural for that smell to dissipate. And even though you farted over there, I smell it over here. That's natural. It will require energy to be able to push that fart back in and, uh, where it came out from. That will require a lot of energy. Maybe not the best example, but hopefully you get what I'm saying. All right, so in the example here is the sodium potassium pump, which is an example of active transport, right? And so sodium Na plus, potassium K plus are moved across the membrane um, in neurons and muscle cells, and they help with uh, many things in activating neurons and activating muscle cells. And so for that to be able to happen, we have to store them up against their concentration gradient. That requires energy to pump them from low concentration to high concentration. Endocytosis also requires energy, right? To be able to take a vesicle inside the cell and bind it uh, with the wall of the plasma membrane, and to have it taken into well, in this case, endocytosis, you're taking part of the plasma membrane surrounding something that's outside the cell, bringing it into the cell. Endocytosis. Another word you'll see for this is phagocytosis. Um, and substance can be moved uh, against their concentration gradients in this way, right? So actually, like, there's something outside the cell that I want. I surround it with the cell's plasma membrane, bring it into the cell, and then now it's inside the cell. The reverse is called exocytosis. A membrane bound substance inside the cell binds with the plasma membrane of the cell, and then it can now escape. It can be released outside of the cell. That all requires energy. And that's the end of our presentation on energy. Have a great day.